The Biochemical Society and Portland Press are pleased to welcome you today to the first webinar in our World Series related to biochemical focus on new topics of relevance in current trends in our research fields today. The topics in our seminar over the course of these webinars will cover state-of-the-art topics and topics of relevance. And today, I'm delighted to welcome you to the first of this webinar series hosted through the journal Clinical Science, the topic being ACE2 friend or foe. Today, we are celebrating 20 years of ACE2. So as we can see, in order to appreciate the relevance of angiotensin-converting enzyme 2 within the realms of the renin-angiotensin system, we need to appreciate that the discovery of renin occurred many years ago in 1898. And since then, there have been enormous discoveries in terms of what constitutes the renin-angiotensin system as we know it today. Of major relevance, as I said, is the discovery of ACE2 in 2000. And today we are celebrating this discovery and the implications with respect to its role in pathophysiological processes, particularly in the cardiovascular system, and of relevance today with respect to the pandemic of COVID-19, the importance of ACE2 as a receptor for coronavirus 2. The remarkable thing about ACE2, and something we have appreciated over the past 20 years, is that it is a multifunctional protein. What do we mean by this? ACE2, by definition, and through its original discovery, was found to be an enzyme, very important in the conversion or the cleavage of angiotensin 2 to angiotensin 1 to 7. We also knew that ACE2 acted as an enzyme, cleaving angiotensin 1 to angiotensin 1 to 9. And these peptide derivatives play an important role in the cardiovascular system. However, more recently, it has become very clear that ACE2 has many other functions, which we term today as non-catalytic functions of ACE2. These non-catalytic functions play an important role, particularly in disease processes, and we will learn a little bit more about this during the course of this webinar, specifically with its respect to ACE2 acting as a receptor in the ACE2 component related to COVID-19. Today, I'm absolutely delighted to have on this panel with me experts in the field. We will hear from Professor Anthony Turner from the University of Leeds, and he's going to tell us about the discovery of ACE2, something which he was intimately involved in. He will also highlight a historical background in terms of ACE2, where we have come from in the past 20 years and where we are today. I will briefly discuss the enzymatic functions of ACE2, and we will finish our presentations today with Professor Michel Barda from the Max Delbruck Center in Germany, who's going to tell us about non-enzymatic functions of ACE2, a field in which he has contributed significantly and certainly advanced our knowledge in terms of the multifunctional nature of ACE2. We will wrap up this webinar, providing some time for questions and answers. And I would just like to highlight to you that you are welcome to submit your questions at any time during the course of the presentations or at the end of the presentations given finally by Professor Michel Barda. So with that, I welcome you today, and we will start with our first presentation given by Professor Anthony Turner.
Thank you, Rhian, and welcome everybody to our ACE2 webinar in which we hope to bring you up to date with the developments of recent years. So let me begin then and re-emphasize what Rhian has mentioned, that the classical renin-angiotensin system pathway we have known about for very many years now, in which the vasoactive peptide angiotensin 2 is produced from the protein angiotensinogen in two proteolytic steps, one by renin and followed by the action of angiotensin-converting enzyme. And Ang2 acts on its various receptors, producing its effects on the cardiovascular system. In this way, of course, ACE regulates blood pressure. Separately, the testicular form of ACE regulates male fertility, and ACE has other roles as well. And as you will appreciate, ACE inhibitors have been very effective in blood pressure control. But ACE also metabolizes other peptides, most notably bradykinin. So where does ACE2 come into this? ACE, as you will have seen, was discovered in the mid-1950s, and since that time has been a major drug target. However, the homologue ACE2, as it became known, remained undiscovered and pretty much unpredicted for almost 50 years. And we set about looking for a homologue for ACE because we knew that peptidases often existed in small families. We had worked on ACE for many years. Another peptidase family, the neprilysin family, has multiple members. So as the human genome sequence was close to completion, we began searching for possible homologues in the genome databases. And in this way, we identified an ACE-like cDNA and cloned and expressed the protein and produced a detailed characterization of the enzyme in 2000. And that was shortly followed by a paper detailing very similar properties for a similar clone isolated by Susan Acton and colleagues at Millennium Pharmaceuticals. In fact, it perhaps was not surprising that ACE2 existed because we knew, particularly from invertebrate biochemist colleagues in Leeds, Elwyn Isaac, that Drosophila was already known to express two ACE-like proteins known as ACE and ANSA. So ACE led us to the discovery of ACE2, and since that time, publications, citations on ACE2 have really risen dramatically with two rapid phases, the first being in 2003, after the discovery that ACE2 was the receptor for the SARS-coronavirus. And now you can see how in 2020, the citations are rising very rapidly again with the identification of ACE2 as the receptor for the SARS-CoV-2 virus. So let's just compare the ACE2 family members. And we have the somatic form of ACE that regulates blood pressure it's a duplicated enzyme with two zinc-binding catalytic sites, whereas the testicular form of ACE is just the C-terminal portion of ACE. However, ACE2 closely resembles the N-terminal domain of somatic ACE, but the C-terminal domain is completely different. It resembles another protein, quite separately, not a peptidase, known as colectrin, which plays a role, amongst other things, in amino acid transport. And you'll hear more about that later. So let's look at the topology of these proteins that we learnt about from our sequencing and structural studies. Both ACE and ACE2 are what we term type 1 integral membrane proteins. That is, they have a small cytoplasmic domain, a single span anchoring transmembrane domain, and then the bulk of the protein, including the active site or sites, face the extracellular space where they can hydrolyze circulating regulatory peptides. In fact, for many years, it's been known that a soluble form of ACE exists in plasma, and that's derived from the membrane-bound form by proteolytic cleavage, and the same is true of ACE2, which can also be shed from the cell surface, in the case of ACE2 by a metalloprotease known as ADAM17. In our initial analysis of the localization of expression of ACE2, we found three major sites of expression: heart, testis, and kidney, very similar to ACE, but we now know from much more extensive studies from many groups that it has a much broader distribution and is particularly abundant on many of epithelial surfaces. It was when we turned to look at the specificity of ACE2 that we saw striking differences from ACE itself. ACE is what we term a carboxy dipeptidase and is able to cleave a dipeptide from the C-terminus of a susceptible peptide substrate like angiotensin-1. However, ACE2 is a zinc metallocarboxypeptidase, so just removes a single amino acid from a susceptible peptide, so in this case converting Ang2 to Ang1 to 7. And it was already known that Ang1 to 7 opposes the actions of Ang2. In addition, other differences were picked up. Firstly, ACE2 doesn't metabolize bradykinin, nor is it inhibited by classical ACE inhibitors. So this gave us a new paradigm really for the RAS, in which angiotensin-1 to 7 became a key player, although its actions were already known about, and it has its own independent receptor, the MAS receptor. The numbers in this diagram here indicate the efficiency of hydrolysis of different peptides by these enzymes, the Kcat over Km. So you can see that ACE2 is actually very efficient in converting Ang2 to Ang1 to 7. If we now look at the structure of these two enzymes, you will see the remarkable similarity of the two proteins. Here we see the structure of the end domain of ACE with the active site and the key binding residues shown here, the zinc ion as a grey ball, and we can mutate that into ACE2 with one click here, and you can see that apart from some minor changes in the active site, the folds of the two proteins are virtually identical, confirming the similarity. We can home in on the active site, where we did quite a bit of mutagenesis studies and modeling studies, and you can see here we have ACE with, again, the zinc ion here and the different key residues of binding subsites, and in comes the substrate, which is then cleaved in a dipeptidase fashion by ACE. We can now look at the changes that exist in ACE2. You can see the alterations there, and we can see how that changes the specificity of the enzyme producing a carboxypeptidase action. So that covers really a lot of what we know about the specificity and activity of ACE2, but the real surprises came 3 years after its discovery, when it was found to be the functional receptor for the SARS virus, and of course this year also for the SARS-CoV-2 virus, and you'll be hearing more about that shortly. But perhaps we shouldn't have been too surprised, because cell surface peptidases commonly act as viral receptors, including coronavirus receptors. So, for example, aminopeptidase N, or CD13, was known over almost 20 years ago to be the receptor for the human coronavirus 229E, and also a receptor for two porcine coronaviruses. And dipeptidyl peptidase 4 is the MERS coronavirus receptor. And as it happens, ACE2 is also a receptor for another coronavirus, NL63. So just to summarize then, ACE2, we believe, is a cardioprotective agent. It counterbalances the actions of ACE in the renin-angiotensin system. And I think you'll see that we now quite well understand the differences at molecular and cellular levels between ACE and ACE2. ACE2 metabolizes a range of peptides, but a different set from those of ACE. But as again you'll hear shortly, ACE2 is a multifunctional protein, not just a zinc peptidase, and with this variety of activities, modulating its expression or activity could provide new therapeutics. So I'd just like to finish by acknowledging those who've contributed over the years, most particularly my two senior colleagues in Leeds, Nigel Hooper, now in Manchester, Dan Lambert, now in Sheffield, and others. For the structural and modelling studies, Ravi Acharya also, and Ed Sturrock, and in Leeds, Elwyn Isaac, who contributed with new information on the Drosophila proteins, and Eric Carron, who at the time was at SmithKline Beecham, who helped us enormously in the very early stages when we were searching for the ACE2 homologue, and our various supporters over the years. Thank you.
Anthony, thank you very much for providing that background on the discovery of ACE2 and the biochemistry and the regulation of ACE2. So I will be talking about the enzymatic functions of ACE2, Anthony having given a very nice overview of the discovery and the biochemistry of ACE2. And as Anthony has just said, ACE2 is an absolutely critical peptidase within many biological systems. But from a very simple point of view, we know that ACE2, acting as a peptidase, binds its substrate angiotensin-2 to generate the product angiotensin 1-7, which in turn has its biological functional role. As was mentioned previously, there are many biological substrates that have been identified for ACE2. And we can see some of the many of these substrates that have actually been identified. But the one that is most important and most potent with respect to substrate enzymatic activity is angiotensin-2, resulting in its cleavage and the production of angiotensin 1-7. Now, as I mentioned, we cannot look at angiotensin converting enzyme 2 and its product angiotensin 1-7 without appreciating its place within the renin-angiotensin system. This is a busy slide, but it really is to highlight to you the complexity of the renin-angiotensin system as we know it today. It wasn't that long ago that we knew that angiotensin 2, the product of angiotensin 1, through cleavage by angiotensin-converting enzyme, was the major vasoactive peptide of the renin-angiotensin system. And for all intents and purposes, angiotensin 2 binding to its AT1 receptor is responsible for most of the physiological and pathophysiological processes that we understand. Angiotensin 2 is a very potent vasopressor peptide. It is profibrotic, pro-inflammatory, hypertrophic, and it also causes retention of sodium, important processes in many cardiovascular diseases. On the other hand, production of angiotensin 1-7 from angiotensin 2 through ACE2 activity and binding of Ang 1-7 to receptor mass has opposite effects for all intents and purposes to activation of the AT1 receptor. We know that Ang 1-7 is antifibrotic, anti-inflammatory, anti-proliferative, antithrombotic, it reduces blood pressure, and it induces natriuresis. And for these reasons, the ACE2 Ang1-7 MAS pathway have been identified as the counter-regulatory axis of the renin-angiotensin system. Now, in order for angiotensin-converting enzyme 2 to induce its biological and functional effects, it obviously needs to be expressed and functionally active in specific cells. And as Anthony mentioned earlier, originally ACE2 expression was thought to be quite restricted to only a few tissues and organs. But we know today, both at the gene level and at the protein level, that the expression of ACE2 is very multi-system and multicellular. And there are many different cell types in different tissues and organs that express ACE2, as depicted and highlighted in this slide over here. I would just like to highlight to you that the expression and activity of ACE2 is really important in terms of physiological and pathological actions. It seems that ACE2 under normal conditions has an age, sex, and race dependency, because ACE2 expression seems to be higher in older people, seems to occur predominantly in males, and seems to be highly expressed in Blacks and Asians. Pathologically, ACE2 expression is increased in many different diseases, for example, heart disease, diabetes, pulmonary hypertension, and even more recently from autopsy specimens and postmortem studies from patients who've died from COVID-19, there seems to be an increase in expression of ACE2 in not only the lungs, but also in the heart. And I just want to highlight to you, with respect to what we understand in terms of ACE2 expression in COVID-19, it actually seems rather unclear at the moment, because experimental models actually suggest that ACE2 expression may be decreased. And there's also been the suggestion that actually there is renin-angiotensin system disequilibrium with a decrease in the ratio of ACE2 to ACE. This is a growing field, and we certainly need to learn a lot more about this to understand the implications of ACE2 expression in COVID. So ACE2, been around now for 20 years, what have we learned with respect to its physiological and pathophysiological relevance? Well, today there are a number of different ACE2 knockout mice that have shown us that there are some phenotypes in some of the experimental conditions, but not in others. And in this study that was undertaken by the Kaufman Laboratory, they actually tried to phenotype using different types of ACE2 knockout mice what actually occurs in physiological basal conditions. And what we can see here is that some groups did identify phenotypes in ACE2 knockout mice, particularly impaired cardiac function, but other groups failed to show any phenotype in ACE2 knockout mice. I want to highlight to you that this was in baseline conditions, and so it does appear that ACE2 has very modest effects in baseline cardiovascular function. However, ACE2 seem to have major effects on disease susceptibility in cardiac, renal disease, as well as in hypertension and in diabetes. And I share with you just a few examples of the many in the literature today to show you that if you do modulate ACE2 in mice, this can bring out an increased susceptibility to disease when you challenge these mice. So for example, in this study by Lisa Cassis's group, she looked at male and female mice that were obese in mice that had ACE2 knockout and mice that had functionally active ACE2. And what we can see in the female obese mice is that their response to angiotensin 2 in terms of blood pressure elevation was markedly increased when ACE2 was deficient. We also know in other studies that ACE2 seems to be dysregulated in the context of diabetes. And here is an example of two different mouse models of diabetes, the DB-DB mice and the streptozotocin-induced mice. And we can see that in these mice, there's an increase in the expression, particularly at the protein level of ACE2, associated with an increase in ACE2 activity. And very interestingly, this is associated with a downregulation of ACE, as we can see in the lower panels. This study by Daniel Batlier's group also showed us that in this DB-DB model, there was a very clear relationship between the expression of angiotensin-converting enzyme and angiotensin-converting enzyme 2 and ACE activity. As we can see in panels B and D, both for ACE and ACE2, there's a linear positive association between the expression levels and ACE expression. Very interestingly, this significant relationship does not seem to hold true for ACE2 messenger RNA and ACE2 activity, as we can see in panel C. Here is another study implicating an important role for ACE2 and Ang127 in terms of their protective effects. In this very interesting study by Reisader's group in Florida, they looked at monocrotaline-induced pulmonary hypertension. And what we can see that in these mice that have got pulmonary hypertension, they have marked cardiovascular fibrosis indicated on the top panel and associated vascular remodeling. When these mice with pulmonary hypertension are treated with ACE2 or with Ang127, the ventricular fibrosis and the vascular remodeling are markedly improved. And so the mechanisms whereby ACE2 and Ang127 may be mediating these protective effects is now being recognized to be occurring via the receptor mass, as Anthony mentioned a little earlier. So what we can see here, similar to the complexity of the renin-angiotensin system that has grown over the past few years, so too have the receptors related to the renin-angiotensin system. But if we just focus on the ACE2 and Ang127 mass axis, we can see that activation of this axis is associated with protective cardiovascular effects. And we, with Robson-Santos, a number of years ago, started to look at the signaling pathways through which angiotensin 127 through mass may be inducing some of these cardiovascular protective actions. And we can see here in vascular cells that if we expose these cells to Ang127, there's an increase in the production of nitric oxide as indicated in the upper panels. And we can see that this is mediated through the MAS receptor because in the presence of A779, the MAS inhibitor, there's an inhibition of Ang127-induced nitric oxide production. And this is associated, as we can see in the lower panel, with an increase in phosphorylation of nitric oxide synthase, an effect that is blocked when we inhibit AKT with wartmenin. We also showed that Ang127 protects against angiotensin 2 signaling in human vascular cells, because here we can see that angiotensin 2 induced activation of ERK and CSARC is markedly attenuated when we pre-treat these cells with Ang127. And so what has now been put together by Robson-Santos and many other groups is the definition of the signaling pathways through which Ang127 through the MAS receptor may mediate some of the cardiovascular protective effects, most importantly through activation of the nitric oxide synthase pathway, activation of nitric oxide, and also production of cyclic AMP. So to finish off then, putting into context for you, the importance of ACE2 as an enzyme, I focused very much on ACE2 and its activation in terms of cleaving angiotensin-2 to produce angiotensin-1 to 7 and thereby induce its cardiovascular protective effects through the MAS receptor. We also know that Ang1 to 9 is produced through ACE2 activation, but we know much less about this. And as I said, production of angiotensin-2 from Ang1 through ACE is responsible for many of the cardiovascular injurious effects. And finally, as was highlighted previously, it's important to appreciate that the therapies that we use today to target the renin-angiotensin system, such as the ACE inhibitors, do not inhibit ACE2. So this is an exciting area in terms of physiology and pathophysiology, because we are always looking for better and improved treatments to address disease. And perhaps by shifting the ACE-ACE2 balance, this might be a new strategy in terms of upregulating ACE2 and thereby promoting the cardiovascular protective effects through an upregulated ACE2 Ang1 to 7 system. And indeed, today, we know that there's a recombinant human ACE2 that is being investigated, ACE2 activators, and also activators of the Ang1 to 7 MAS pathway. So the take-home messages for today relate to ACE2 is a very important enzyme as part of the renin-angiotensin system. ACE2 has multiple substrates, but Ang2 is probably the most important as we know it today. The ACE2 Ang1 to 7 constitutes the counter-regulatory axis of the renin-angiotensin system. ACE2 is widely expressed and seems to be linked to ACE, sex, and race. ACE2 has modest effects on baseline cardiovascular function, but has major effects on disease susceptibility. There are now therapeutic strategies that are being developed to upregulate ACE2, and some of these are under development for clinical trials. And now we will continue and advance the field of ACE2 by looking at some of the other functions that are not related to the peptidase activation of this protein. And I will now hand over to Professor Barda, who will talk to us about the non-enzymatic functions of ACE2. And with that, I'm absolutely delighted to welcome Professor Michelle Barda from the Max Delbruck Center. And Michelle's going to talk to us now about the very recently identified non-enzymatic functions of ACE2. Michelle?
Thank you very much, Rhian. And thank you for organizing this webinar, and welcome to everybody who is here to hear my talk. 1 second. So now you should see my presentation. I will talk about the non-enzymatic functions of ACE2. And you have seen already this kind of cartoon by Anthony showing you that this strange molecule has two completely independent parts which have been put together in evolution. I think no one knows why. One is the carboxypeptidase part, about which Rhian has already talked, which is essential for the production of angiotensin 1 to 7. And there's a collectoring domain, which is important for another function I will talk first about. This is the function as an amino acid transporter. So what is the situation there? There is a transporter of a lot of neutral amino acids called B081 or SLC6A19. And this molecule needs ACE2 to be expressed in the cells where it is necessary to be expressed. And this is in intestine, and it needs another molecule in the kidney where it's also very highly expressed, and that's collectorin alone. So this homology protein to this part of ACE2, collectorin, has the same function in the kidney. And I think I can show you the original data which have shown that, and this is this paper here from 2009. You can see in a normal animal on this side here, you see ACE2 being expressed in the enterocytes of the gut of the small intestine. Also B081 is there, and they merge well together. So really consistent with the idea that the two are really in a tight complex. In the ACE2 knockout, obviously there's no ACE2 as it should be, but there's also no B081 whatsoever in this intestine. So the whole expression of this protein is depending on ACE2, obviously, because it's probably degraded when it's not placed in the right position on the plasma membrane. So here's a Western blot showing the very same thing. In normal animals, you see ACE2 being there in the small intestine and the kidney. In an ACE2 knockout, obviously there's no ACE2, neither in kidney nor in small intestine. That's what to expect. Here's B081, and it looks like the knockout of ACE2. There's absolutely nothing there in the intestine of ACE2 knockouts. There's no B081. But there are normal levels of the same molecule in the kidney in the ACE2 knockout. There, ACE2 is completely unnecessary. Here, you need colectrin. So that's colectrin knockout. No effect in intestine. There is ACE2. But in kidney, there's no colectrin, and that's why there's no B081. What is the consequence of this lack of this transporter? It's not the only transporter in the intestine which transports amino acids, so there's redundancy, so there's not much effect on most amino acids. So in the knockouts phase 2, you see a reduction in glycine, which is modest, but you see a quite strong reduction in tryptophan. And that, I think, is a major effect which this has on amino acid metabolism of a mouse. And when we saw that, we immediately got the idea what happens with another very important molecule in ACE2 knockout mice, which depends on the presence of serotonin, of tryptophan, which is serotonin. So serotonin, as you may know, is produced by two enzymes TPH, hydroxylase enzymes, one in the gut, one in the brain, TPH1, TPH2, and both produce an intermediate, which then is further processed to the final 5-HT, the final serotonin. So we checked first, do we also find in our ACE2 knockouts, which we got from Joseph Penninger, the same kind of reduction in tryptophan, and we could reduce exactly what was published. In blood, it was reduced by more than 2/3, and in the brain, we have got more than half reduction of tryptophan. This would mean we would expect to have reduction in serotonin. So we checked that, and it's true. In the blood, we have about half of serotonin. In the brain, it's about 30% less than normal. And so we checked the animals in behaviors and so on. I'll only show you one thing. This reduction in serotonin probably is involved in one of the effects we see. If you let the mice run in a running wheel, normally a mouse would increase neurogenesis in the hippocampus. So this is the wild type here. And you see there's a clear increase by more than double of the cells which divide in the hippocampus. But this effect is completely gone in the ACE2 knockout, and it's maybe due, we cannot prove it yet for sure, on this effect on the serotonin level, because you know serotonin is for this also an essential mediator. So this is now the summary of the first part of the amino acid transporter part. Sorry, there's an error here. ACE2 is essential for the functional expression of B081 as amino acid transporter, but only in the intestine, not in the kidney. ACE2 deficient mice have therefore reduced TRP levels and also serotonin levels in blood and brain. And probably because of that, the exercise-induced increase in hippocampal neurogenesis is abolished in these mice. So this makes ACE2 a novel factor essential for serotonin generation and requires and being required for the exercise-dependent modulation of adult neurogenesis, a function I think nobody would have predicted before. But now comes the most sort of fancy function in the very moment of this molecule, which is the binding of this COVID-19 virus SARS-CoV-2. And there are two areas which have been found by crystallography studies. One is far away from the active site. One is overlapping with a very border of the active site, where the virus obviously binds to ACE2. So we have started to work on that also when COVID-19 came, especially to find molecules which could interfere with this binding. I can't show you any molecule in a moment, but I can show you at least one assay which we have established to study that. What we have done, we have transfected HEK cells, so just normal kidney cells, with ACE2, and you see here there are some but not all cells infected by ACE2. And then we gave a receptor binding domain of the SARS-CoV-2 virus linked to a His tag on these cells and let it bind. And you see when you bind them, exactly the cells which have ACE2 also bind the RBD and the others don't. So you can see this merge here. So clearly we could confirm that that ACE2 is the binding factor of the spike protein of the SARS virus. So how does it happen? This is a cartoon from a paper of the group of Pöhlmann in Göttingen, where it summarizes sort of what is known up to now, how this binding happens and who is involved in this interaction. So first there's the virus, can be COV1 or COV2, both look more or less the same and both bind with the spike protein to ACE2. That's only one aspect. There's a second very important molecule, TMPRSS2, another protease, which is sort of very closely associated to ACE2. And only if it's there, then this attachment of the virus leads to the cutting of the viral spike protein by TMPRSS2. And this cutting is essential for the uptake of the virus into the cell. If it doesn't happen, the virus cannot infect the cell. So this triangle of these three molecules here is important for functional uptake. And there were some interesting data, especially in the SARS-CoV-1 times of these two first papers here, which showed that this TMPRSS2 does not only cut the spike protein, but also cuts ACE2. And this was sort of counterintuitive, because if you cut off ACE2, sort of liberated from the cell membrane, that's what TMPRSS2 is doing, then you would expect that the whole complex would be released from the cell and you would not infect it anymore. But the opposite is the case. In these papers, they even say you need this cut of ACE2 to have an effective uptake of the virus into the cell. As far as I know, it has not been shown yet for COV2, so it's just for COV1 yet. So this was at this time, I think, completely unexplained how this can happen, that you cut this receptor off and get a better infection. Now I think we may have an idea why this happens, because now we have a lot of new data. I will show you in a minute. But just one thing which is sort of coming from this study is that there is a chance to inhibit the infection by inhibiting this very crucial enzyme for it. And there are two drugs already on the market in Japan, at least. They are approved for pancreatitis. They inhibit this enzyme. They're in the moment in clinical trials. I haven't seen the data, but I think this is a very, very promising new target, which may solve the problem that we finally get a drug against this virus. But now I come to the new findings, which also gave a lot of new insights into ACE2 itself. So this is a study where they did cryo-electron structural studies on a complex of ACE2 with B081 and the viral binding protein. So all three are together. I don't know why these authors did this in a triple complex, because it doesn't really happen in nature very often. This is happening only in the intestine, not in the lung. This B081 with ACE2 is not existing in the lung. So it's probably not what the virus sees when it enters the host. But anyhow, it shows us one important thing. It shows us that ACE is a dimer. This was completely, I think, unknown before. Nobody thought about that. But in this structure, it's a clear dimer. And this dimer is independent of the binding to B081. So this part is separate. Here's the binding site between B081 and ACE2. And the dimerization is purely between ACE2. So really, the ACE2 forms a dimer. And this may now explain the story with TMPRSS2. If TMPRSS2 is cutting only one of the dimer, maybe the whole thing gets a new conformation, which allows the virus to enter the cell. But this is pure speculation. I think there's no data available. So here's the dimer in even more details. Maybe it doesn't make sense to go detailed in that, but at least you could exactly show which are the dimerizing molecules, and can now study in the future how important they are for normal function of ACE2. The second important thing which was studied was the expression of ACE2, especially in human cells and human tissues. And obviously, it was important to see which cell type in the lung is the most sensitive one. And the only cells, you can hardly see it maybe here, which express in the lung, and also only a few of them is the 82 subtype of pneumocytes. And that's, I think, the most important cell concerning the infection, at least in the lung, where the worst consequences are seen. You see TMPRSS2 is also there, even more in the same cell type, so they really are co-expressed, and that makes this cell so susceptible to the ACE2 infections. Here's a more general overview, in this case of monkey, but closest to human at least, where a lot of different tissues have been checked. And again, as partially shown already in rodents. The gut is the most expressing site also in the monkey. Here, unum, ileum. Lung is not very much. Very few cells express it, but it's enough to get this strong phenotype of the disease. So another interesting cell type, which was studied in this paper here, is the susceptible cells in the nose. And this is the expression by an antibody here in these cells. Here's a schematic. And this is the cell, probably the cell which is important for smelling and yeah, to have olfactory sensation. And as you may know, one of the main side effects of coronavirus infection is loss of this, at least for some time. And this group thinks that this is because of these cells are getting inactive, and maybe the olfactory epithelial neurons are not anymore supported by these cells anymore, and that's why they are not able to wire correctly and give the sensation. The other thing which came out, which is also a completely new finding, is that ACE2 is an interferon inducible gene. So here in this paper they showed that if you give to certain primary cells from nasal scrapes from humans, I think in each case a single person, and you give increasing amounts of interferons, you see a clear increase even to 5 times or even more of ACE2 expression in these cells. It's a very specific primary cells. I wouldn't say this is already the final word, but at least there is also interferon activation, which we don't know what it means for the COVID-19 infection, but maybe this also should be taken into account, that this is a protein which reacts on viruses whatsoever. So the summary of this part is ACE2 is bound by the SARS-CoV-2 spike protein. That is the reason why we have this disease. ACE2 expressed in cell types, the 82 cells in humus, in the lung, testicular cells in the nose, enterocytes, vascular pericytes, and so on, are main targets for SARS-CoV-2 infection. And just to conclude, ACE2 is hijacked by SARS-CoV-2 for host cell entry, and therefore the interaction of ACE2 with its spike protein is a promising therapeutic target for COVID-19, and we are working on that, as a lot of other people do, to find substances which interact at the site where COVID-19 or the virus is binding to ACE2. So here are the people who are involved in this study. People in my lab and also in other labs of the MDC have teamed up to find these drugs together with people at the Robert Koch Institute in Berlin, virologists. We got the mice for our mouse studies from Joseph Penninger, and a lot of studies I couldn't talk about on the enzymatic functions of ACE2, on angiotensin-2 and so on. We do with Luisa Rabello and Robson Santos in Brazil. Thank you very much for your attention.
So thank you very much, Michelle. We are just waiting for Anthony. And I would like to encourage you, please, to ask your questions by typing them into the question box. We have got about 7 or 8 minutes to answer your questions. And indeed, the questions are coming in furiously now. And I will start asking some of the questions. I will start with a question for you, Tony. There is a question here: does the ACE2 still have activity in zinc deficiency? And do you think that the non-catalytic effects of ACE2 are affected by this? And does this have any impact on non-enzymatic effects? In other words, the dependency on zinc?
Unless there is very severe deficiency, it's unlikely that it would affect the activity of the enzyme, and the same would be true of ACE as well and other zinc peptidases, but it might well be of relevance to some of the other properties of the protein.
Okay, and thank you, Anthony. We now have a question for Michael. Could higher levels of the shed ACE2 binding and neutralization of COVID in blood prevent it from being internalized?
Yes, I think this is a very, very important new strategy to fight COVID-19. There's a company, Apeiron, which you may know. They have recombinant human ACE2 since long time in their on clinical trials or even approved. And they have started a trial also on COVID-19 patients. And they have shown also in a Cell paper quite recently that it works on organoids. So they infected organoids, human organoids, by the virus and could inhibit infection by giving this recombinant human ACE2. So in this setting, it works. The problem is to get it at the right place. That's my opinion. If it's in the blood, I think no problem. You can block it in the blood. But I can hardly imagine how to get the recombinant ACE2 into the lung at the alveoli. That is the problem. So you may be able to to protect from infections of internal organs, but the primary infection in the lung will be difficult to fight with this large protein. Let's see the clinical trial to show us.
And just continuing on that topic of potential therapies, here is another question again targeted to you. Is there any inhibitor of colectrin that is being investigated for ACE2 in the context of COVID?
Not that I know. I don't think any inhibitors existing of this molecule because there's no real activity. It has spines just to the amino acid transporter. And I would not predict that this would make a big effect on COVID-19 binding, but this is just speculation. Because there's normally no P081 there in the lung, so this is not really relevant for the virus. I don't think this COVID colectrin part is important for the virus, but it's really speculation.
Okay. Here is another question related also to the gut. That ACE2 is highly expressed in the small intestine, as we've seen across a lot of species. Regarding the data of COVID-19, there's also a small number of patients with gastrointestinal symptoms. Is there an association? And we do know that the virus can be detected in stools, for example, and I think particularly in children. So I would imagine that ACE2 being in the gut might have some relevance with respect to infectivity in the gut and gastrointestinal symptoms. But do you or Michael or Anthony have any comments with respect to ACE2 in the gut and the potential role in gastrointestinal symptoms in COVID?
There's this paper by Penninger who used also gut organoids, as far as I remember, and he saw infection. But the question is all the time, does the virus reach the right compartment? So this ACE2 is probably showing into the lumen of the gut. And there is some virus, but I don't know whether it's still effective there because there are so much digestive enzymes and so on. So I think it is quite likely that all the gastrointestinal effects are mediated by this ACE2 on the enterocytes, but it's not so easy that it's just when it's in the gut lumen somehow it can be still infected. It must come from the blood somehow and then get anyhow to the ACE2 and the enterocytes. So it's likely to be involved, but it's not sure yet, I would say.
Okay, thank you. Anthony, we have a question for you. The question is ACE N-terminal similar to ACE2? If it's similar, then why does the ACE inhibitor not work for ACE2? Why is ACE2 not sensitive to ACE inhibitors?
Although, as I showed, the similarity between the overall sequence and structures of the proteins is very high, there are the subtle differences in the active site region where substrate binds and also affecting inhibitor binding. So there are amino acid critical changes in the active site that prevent the binding of the ACE inhibitors to ACE2. And we have modeled that in some detail.
Okay, and here is another question for anybody on the panel. The question that has been put forward is, do you know if ACE2 is expressed in the brain and in neural tissue?
It certainly is expressed in the brain. It wasn't something that was initially picked up, but Eric Lazartigue's group in the States have particularly worked on brain ACE2, and it does play a role in central cardiovascular function.
Perfect, thank you.
I also say this is one of the major enzymes which degrades angiotensin-2 in the brain, I would say.
Okay. Our time is running short, but maybe we've got time for one more question for Michael. To what extent does binding of SARS-CoV-2 to ACE2 block ACE2 activity and Ang127 formation?
Very good question. And there has been data also hidden in the literature about SARS-CoV-1 showing that the binding of the virus does not at all affect the enzymatic function. And the other way around, if you block the enzymatic function with MLN 4760, the virus can anyhow bind. So this is completely independent. And we have proven that also with a very short experiment, also with SARS-CoV-2, that still ACE2 is active in the presence of the virus. But yeah, it's not yet published, but it's the same as CoV-1.
Okay. I see that it is now 3 o'clock, which is the end of our webinar. And again, I do want to thank my co-panelists for joining me today. And I really want to highlight that this first webinar that is sponsored through Portland Press and the Biochemical Society, and especially the journal Clinical Science, I would like to highlight that Anthony and Michael have very kindly agreed to being guest editors for a special issue entitled ACE2, a multifunctional protein from cardiovascular regulation to COVID-19. I encourage you all to please submit your best science to this issue if it does relate to ACE2, and you can contact the journal or Portland Press at the website as indicated here. So with that, again, a great thanks to our co-panelists, and please join us next time for the next webinar, Building Gene Drives to Beat Malaria, on the 11th of June at 3 o'clock, chaired by Professor Sheila Graham. Thank you.
Thank you.
Thank you.